This lesson's an introduction to genetics and genetic terminology. So this is a drawing of a chromosome. I'm drawing it with one chromatid, but I could have drawn it after DNA replication where it would have two sister chromatids. And the first word to know is locus. And a locus is just a location on a chromosome. It's like an address on a chromosome. And at that location, we would have a gene. Now, a gene is a unit of heredity. It's the smallest possible unit of heredity we can talk about. And it can be helpful to know that we knew that there were genes, particles or units of heredity that were passed from parents to offspring before we knew that DNA existed and that genes were made of DNA. But now we know that DNA is what makes up genes, and we can think of a gene alternatively as being a stretch of DNA that leads to or codes for a functional product. And that functional product is often a protein, but it could also be an RNA. RNA has functions in the cell, as we know from making up ribosomes, for example. So a gene is a unit of heredity or a stretch of DNA that leads to or codes for a functional product. So here's part of the chromosome. Remember, the chromosome is a single long linear double helix. So this might be our gene. And there are different versions of that gene that we might have in the population. I'll label them like this. So this is the A version, the B version, the C version, and those different versions we call alleles. So an allele is a version of a gene. And again, it's important to remember that there can be many more than two in a population because we think of individuals, and an individual can only have two alleles because an individual only has two copies of one chromosome in the diploid state, which we'll talk about in a moment. We can often forget that there can be many more versions of that allele in the population. So that's locus, gene, and allele. Now, a genotype refers to the particular genetic makeup of an organism, and so you can think of that as being all of its alleles, whereas a phenotype is the observable characteristics of an organism. So that might be what it looks like, what its physiology is like, its behavior, its development, all of that is part of its phenotype. So genotype is a genetic makeup, whereas the phenotype is the observable characteristics. So for example, at one locus, the genotype might be big A, little a, let's say, and the phenotype might be that the individual has brown hair. Next terms to know about are diploid and haploid. Diploid means two complete sets of chromosomes, whereas haploid means one set. So for an organism with just one chromosome, the diploid state might look like this. I'll draw blue and red for the two different chromosomes, one from mom and one from dad, whereas the haploid state might look like this. And here's another version with the chromosome from the other parent. And when we talk about meiosis, we will talk about the process of going from a diploid state to a haploid state, because this needs to be done in making sex cells, sperm and egg cells, so that they can recombine during fertilization and recreate the diploid state. So diploid is two sets of chromosomes and haploid is one. And you'll see numbers associated with this written in this fashion. 2n equals 46. This would be for humans. 2n means we're in the diploid state, and 46 is the total number of chromosomes in the diploid state. Whereas this, n is equal to 23, the n without the 2 in front of it, 1n, refers to the haploid state, and this means we have 23 chromosomes in the haploid state. So 2n equals 46, the diploid state has 46 chromosomes in humans, and n is equal to 23 means the haploid state has 23 chromosomes in humans. Now, when you have a diploid organism or a diploid cell, you've got matching pairs of chromosomes like this, one from the mom and one from the dad. So we call those matching pairs homologous chromosomes. Homologous chromosomes are matching pairs of chromosomes in a diploid cell. Again, one from the maternal organism and one from the paternal organism. And when we've got matching pairs, we can categorize them as homozygous or heterozygous based on the alleles at a particular gene. So if we look at a particular gene and the alleles are the same, we call that homozygous. And if we look at a particular gene and the alleles are different, we call that heterozygous for that gene or for that locus. Using what we did above here, A, let's say the gene is called A, we might have capital A, or little a, whereas heterozygous would be one big A and one little a. Or if we had above G with a superscript of A, this might be 1, and this would be heterozygous. So that's a quick introduction to terminology in genetics.